Hey, more the merrier. Okay, we're back at EOTech 2024. My name is Ben Kinishenko. I'm moderating the wastewater stream. And this afternoon, we have Shawn Dubois from IDEXX Tecta. Okay, IDEXX Laboratories, thank you. Shawn's a senior scientist. He has been part of the water business for over 20 years, working in research and development, and his current role managing the product support group for North America. In addition, Shawn manages the ISO 17025 2017 accreditation for the Water Quality Control Lab. Shawn has a BA in biology and a BS in environmental science. He's a participant in several standards development organizations, including standard methods for the examination of water and wastewater. That's our Bible, hey Mike? Interestingly, this year, it was a long weekend. We had a water break. We were able to collect samples. But the our contract lab wasn't open. This is up in Squamish. But our health authority, our drinking water officer, had just been trained in the IDEXX system, and they had it on site, so they were able to run samples for us. Nice. So helped us out in a bind. It was great. So welcome, Shawn Dubois.
Thank you. All right. All right. Everybody hear me okay? Perfect. Okay. So thank you, everybody here and virtual, for joining today. So I'm going to go over the newest technology in IDEXX for the water testing business. And this is Tecta for the automated bacterial testing of water samples. So kind of a brief objective for today. Going to go over briefly introduction to water microbiology, why we're doing the testing that we're doing, going over the Tecta instrument itself and the associated reagents for testing, basically how to run it, and give us some real-world examples of how this system could be used out there. And then have a few minutes at the end for any questions and answers. Well, not for your answers, my answers for you. So starting off again, intro to micro. My training again is actually in marine biology and marine ecology. So I've learned micro over the years in my position, and really can kind of sum it up in the far side. Does a great job of taking science and making fun of it. So I thought this is a great way to kind of start off. There's a little bit of humor. Thankfully, we will not be talking at all today about petri dishes or colony of bacteria. So we're going to focus more on automation that makes it nice and simple. So we'll stay away from that. So starting off, where did the bacteria come from and why are we looking for them? So bacteria can come from a lot of different sources. The ones that we care about when we talk about water testing can come from different locations, such as soils, off of plants, naturally occurring in the water. But the ones that we tend to focus on the most are ones that are coming from fecal sources. And those fecal sources can be just any kind of animal that's out in the environment. And of course, the big one for us, humans. This is a picture of my daughter from, at this point, almost 5 years ago, waiting for a family of ducks to make their way into the pond, so that way she can get into her kayak and head out. But yeah, this is a great image. It just kind of shows like all the different sources of bacteria that we'd be looking for. So the next question is, why are we looking for them? So when we talk about water that we're either going to use for consumption, use for recreation, or in the case of wastewater, what we're going to be putting out into the environment, the reason why we're looking for bacteria is as a surrogate for the actual things that we care about. And the things we care about are viruses, pathogenic bacteria, amoebas, just other parasites. So there's a lot of them that could be out there. There's a lot of them that can show up, especially in waste, and they can be very difficult to find. It's not very cost effective to look for them, nor is it a quick and easy test. So rather than spend all that time and energy looking for every possible thing that could be out there, we're going to look for something that acts as a surrogate that basically tells us whether or not something could be present. And that's the concept of the indicator organism. So think of it another way, rather than looking for fire, we're going to look for smoke. We're going to look for something that's very easy to find, and if we find it, then that tells us that we might have a problem. So when we talk about indicator organisms, we want something that would be present when a pathogen would be present. And the other side of that, we want them not to be around if there is no contamination in the water. We want them to be found in high numbers, even more so than how much pathogens can be in there, because we're going to take basically a 100 ml sample that is representative of millions of gallons of water. So we need to have something that we can easily find in a very small sample size. We want something that's going to survive very well in the water. We want to be able to test water throughout the process and know that that indicator organism is going to still be present regardless of how much time it takes for us to collect or between collection and actually running the sample. And now when it comes to the work that's actually done in a laboratory setting or at the treatment plants, we want something that's easy and safe for people to work with. We don't want to put people at risk by having to grow up things that could actually make them ill. We want results that are going to be rapid. We want to be able to make decisions quickly, and so we can move forward with any kind of remediation steps that we need to do. We want something that's relatively inexpensive. When we talk about some of the tests that are out there for different bacteria or viruses, it can be very costly. So we want something that's going to be on the lower cost side. And the big one is accuracy. We want to be able to say with confidence when we are getting our result from our test that the results are true and that we are making the right decision, whether that be to say that the water is safe for people to use or that there needs to be shut down and then some fixes. And one of the other things I should focus on too is also the easy part. And the reason why we want something to be easy is because we want to be able to let anybody run the test. You shouldn't have to be a trained microbiologist in order to run these tests. We want something that's going to be applicable for kind of anybody to run. Because the more people that can run it, the more likely we are to test. And the more likely we are to test, the more likely we are to catch things. All right, now I'm getting to the boring part of microbiology. Growth curves, yay. So when we're talking about bacteria, we're talking about a bacteria that has gone through some kind of treatment process, whether it's been chlorination, ozonation, UV treatment, whether it's just naturally occurring exposed to sunlight or mechanically. And what we're going to do is we're going to put them into our test. And what we're going to give them is everything they need to become healthy again. When they're injured, their cells are, the cell membrane is usually pretty broken. They're actually leaking stuff out into the environment. They're not very healthy. They're alive, but they're not really doing much. So by taking them and putting them in our tests, what we're doing is we're giving them everything they need to become healthy again. And that's this first part of the graph right here. And this is known as the lag phase. And basically what's happening is the bacteria is taking all the nutrients that we've given it, repairing itself, becoming healthy, and growing. And bacteria, the way that they multiply is by division. So as a cell becomes healthy, gets large, it will then split, and that one cell will become 2. Those 2 cells go through the same process, they become 4, and basically we double every X amount of time. And that's where we get the log phase. Now when we talk about any kind of our different tests that are out there in order to find bacteria, these 2 phases are the ones that we focus on the most because this is what is going to tell us when our test is complete and that we can say whether or not the bacteria we're looking for are present. There are 2 more phases that we're not really going to focus on, but just for information-wise, we have a stationary phase. This is where bacteria are still growing, they're still dividing, but now we also have bacteria that are dying off. So the population is kind of peaked. It's not getting any bigger, but it's not really shrinking. And then finally, we have the death phase. And this is where the amount of bacteria that are dying are outnumbering the ones that are new. So the population is basically just decreasing. But again, for us, when we talk about detection, we're focusing on the lag and the log phase. So, anybody out there can tell me something, a recent event in the news that focused heavily on water quality, maybe specifically with E. coli and enterococci? How about the Summer Olympics? The river in Paris was exactly, not exactly the best location for swimming. For years, Paris has been working on fixing their system enough to allow some of the events to happen in the river. To some degree, it worked. however, not completely. So we heard a lot leading up and through the Olympics up until the day of the events about how they were having way too much E. coli in the water, way too much enterococci, and they were able to get it down to levels that were deemed safe by Europe, which is, I will say, about 10 times higher than what we usually say is safe in here in North America. So they were saying anywhere between 1,000 and 1,500 bacteria like E. coli or enterococci per 100 ml was okay. Whereas here it's mostly 100, 150 is like the upper limit. And then you might have heard some of the fun things that people were doing in order to be safe as they were swimming in there, including drinking excessive amounts of soda to make their stomach acid more acidic to hopefully kill off anything they might ingest. Some people were saying that they stopped washing their hands for weeks before the Olympics to make sure they could build up a tolerance. Again, a lot of these were all just not going to help. Because again, people are focused on the E. coli and the enterococci numbers and saying that's what they were concerned about. But the real concern is that what those represent and that there is other things in there. There's viruses. And if there's really high bacteria, there's also a high probability of viruses and other pathogens, which some of the athletes came out of the water and they were not feeling too well for a little bit after. So, but anyway, real world situation, big news, all over the planet showing off. So, okay, that's it. That's all microbiology. Now we're going to talk about the actual instrument. So, what is IDEXX Tecta and what does it do? So, it is a new technology for us. It is a rapid automated system for the detection of coliforms, E. coli, fecal coliforms, and enterococci in different water matrices. And basically, the machine is going to do a majority of the work for you, and it's going to allow basically testing to be done basically anytime by anyone and anywhere, kind of what we kind of want for testing. We want to be able to make sure that anybody can do it. Here we have a picture of two different instruments. Here's a little bit more information. Again, high level, there is the Tecta B4. The B4 allows for 4 tests to be run at one time. It has a small footprint. It's good for like local, small locations or remote, or if you're only testing a few samples here and there, then a B4 is a good size. The B16 allows you to do up to 16 samples at one time. And it can be actually, we have different tests, and certain combinations can be done at the same time. So you can actually use this to run multiple tests at the same time. The other thing about this way the system works is the counting range of what it can give you for quantification is less than 1, so basically 0, up to 10 to the 8 per 100 ml. So a very wide counting range with no dilutions required. And as for the tests, there are 4 different tests. The big one is the Tectalert ECTC, which is for the detection of E. coli and total coliforms, incubated at 35 degrees. And this one in the United States, the US EPA has approved for drinking water as a presence-absence test. And I'll be very upfront right now. If you are wondering about any kind of approvals, that's the only approval that we currently have, truthfully, for Tectalert and Tecta. So a lot of the people who are using these products are using them for non-compliance. They're using them for their on-site testing, monitoring, and then have their accredited method that's being done as their actual answer. But there's people using this system in order to do their own monitoring for non-compliance. So the next test is Tectalert EC. It's an E. coli only test, slightly different in that we're only looking for E. coli, has a slightly higher incubation temperature. There is Tectalert FC for fecal coliforms. Again, going by the definition of what a fecal coliform is, it's a coliform that can grow at 44.5. So that's what the Tecta will incubate at. And then Tecta ENT, Tectalert ENT, which is for enterococci. So we'll get to the instrument in a minute, but first the other part of the technology is the actual Tectalert cartridge. So the cartridge has several key things. Nicely labeled. We'll have the lot information right on there, so therefore, if you separate it out from the main kit, you still have the traceability as to what you're using. Has a space that allows you to put any kind of label information that you have for your sample. There is tamper evident tab along with a little button here that basically will let you know whether or not that test has been opened or compromised in any way. There is an embossed 100 ml line that you can use for actually measuring out your sample within the cartridge, or you can pre-measure it out and use that. The cartridge itself is made of polypropylene, and then the very unique part of this cartridge is this polymer partition that's actually embedded into the polypropylene cartridge here at the bottom. You can kind of see this like little nub. So this is actually part of the technology and how this test works. Seems very simple, but it's very important. And then finally, all the reagents are actually contained inside the cartridge for that sample, so there's no separate container that you need to then add into the cartridge. It's all contained into this one device. So the way this works is actually very similar to a lot of our other enzymatic methods. Who here either uses or knows about Tectalert or Tectalert 18? Okay, a few of you. Works on that same technology. And basically what we're doing is we're looking for specific enzymes that our target bacteria have. And really simply how it breaks down is that we have nutrient indicators, which is very simply a nutrient and bound to it is an indicator. And then when an enzyme is present by the bacteria that we're looking for, it basically eats the nutrient, breaks that bond, and the indicator is released. Now, when we talk about Tectalert, which is used for coliform and E. coli testing, there are two different nutrient indicators looking for two specific enzymes, one for coliforms and then one for E. coli. So in the case of Tectalert 18, what's happening is a coliform or E. coli has this enzyme. It will produce a yellow color as the indicator. And then E. coli has a second one that would use the other indicator that will turn that yellow and also make it fluoresce. So what the Tecta technology is doing exactly the same thing. We just have different indicators, but we're still looking for those same enzymes that define what a coliform and what an E. coli is. And basically, you kind of see here a nice little cartoony representation is that when the sample and the reagent are mixed, the substrates are hydrophilic, which basically means that they are water-loving. So it is flowing freely inside that sample mixture. When the enzyme, if the target bacteria are present, cleaves off and breaks off that indicator changes and it becomes hydrophobic, which basically means it wants to get out of water. And what it does is it's trying to move to a location that it's more favorable for it. And that's where that little partition at the bottom comes into play. Basically, that hydrophobic indicator will now be drawn to this area down here and it collects. And here, it will collect, and this is where the instrument part comes in. So the instrument has UV LEDs that shine up into here. As the indicator collects, it gives off more and more fluorescence. The Tecta instrument has spectrometers that look for that fluorescence, and once it sees enough of it, it triggers it, and it says we have a positive result. So kind of going back to what we talked about earlier, the way that this test works is that we want to have bacteria, we want it to be very active, and we want a lot of it. So the more bacteria we have, the more of that enzyme that we're looking for is present. The more of the enzyme we have, the more the nutrient indicator gets used, which means we have more indicator released. More indicator means it collects into that partition, which means that we'll have detection. So going back to earlier, we talked about that growth curve. The vertical axis here, we have the number of bacteria that are growing. So as this goes up, we have more and more bacteria, and then we graph that along time. So the longer we go, the more bacteria we get. Now, if we look at the Tecta and what it looks for when it looks at the fluorescence of the sample, we see a very similar shaped curve form. So over here is the amount of fluorescence that is being detected by the machine over time. So we can see as more time, we get more of fluorescence until it peaks up here. So very similar to what we see for a growth curve for bacteria. And this is actually how the Tecta says whether or not a sample is turned positive. So here's another example of that same kind of that look, where we're looking at the amount of fluorescence that's being formed over the amount of time. And what we can do is basically take that information, look for this point where that curve forms, and that signals the machine to say that the sample is positive. So here's 4 different inoculations of the same bacteria, aimed at increasing or decreasing concentrations of that bacteria. So if we start off high, we see this yellow, 10,000 CFU per 100 ml. We can see that inflection point showing about 9 or 10 hours. That's why that turned positive. If we add a tenth of that amount of bacteria, it's about 2 hours slower, and we get a positive here, 100 CFU here, and 10 CFU here. So what we can do is look at the point that it turns positive based on time and relate that time to the actual amount of bacteria that's in there, the estimation of how much is in there. So the faster we get a signal, that means we have more bacteria. It goes back to this whole idea of more bacteria means more enzyme, more product, and therefore more signal. So what we can do is correlate the amount of time it takes to turn positive to about how much bacteria is in that sample. And this is how the quantification works for Tecta. Technology also has a couple of other benefits. One of the ones that we tend to hear about is human interpretation, especially when it comes to fluorescence or color. Sometimes there can be difficulty in reading some samples, and there's a little subjectivity. Basically means you have to either incubate a little bit longer, see if color or fluorescence develops a little bit more. But with the Tecta, because you're looking at a concentrated signal right at a certain point, and it's all automated by a spectrometer, it gets rid of that human interpretation. Other problem, especially when we talk about wastewater or certain type of waters, that you might have a lot of color, you might have a lot of turbidity that makes it very difficult to read, especially when we're talking about the human eye looking through it and trying to see either a color change or fluorescence. Again, that's eliminated with the Tecta because that partition that's at the bottom is going to be looking for fluorescence in a concentrated spot outside of the actual sample. So it doesn't have the issues with color or turbidity for most samples. The other thing too is that the instrument is consistently incubating the sample throughout the time point, and it's also reading the sample at the same time, which basically means that results are not, you don't have to wait to an endpoint. You don't have to wait 18 or 24 hours to get your final result. The machine is going to do all that work, and it will constantly read it, and it'll give you detection time when it turns positive, and then stop the test. So for the ECTC test, if you have a lot of bacteria, you'll get a result as early as 2 hours. If you have very low counts or no counts, the test terminates at 18 hours. So basically, you'll get a result between 2 and 18 hours, depending on how much bacteria is present or none. Again, because we are not doing the counting ourselves, we're basing the concentration on the time that it becomes positive, means that you can have a much larger counting range without having to do any kind of dilutions. Again, so basically 0 to 1 to up to 10 to the 8 CFU per mil, and no dilutions required. Another benefit of the machine is that as it is running the sample, it is collecting the results for you, and it saves the results. So it gets rid of the human error that can occur by not transferring over the data, missing information such as temperatures or the time that you're supposed to read the result. So the machine takes care of all that for you. It saves that information directly onto the instrument itself, and then that instrument data can be pulled off with a USB stick. The machine can also be set up for email, and basically, besides saving on the internally, it can also send out an email with the results, so that way you can have it as soon as it turns positive. So in a case of water main breaks and all that, where you need to have the answer as quickly as possible, if there's an issue, it can tell you that as soon as it's completed, so you can have results early, so that way you can go ahead and continue on with your actions as opposed to having to wait the 18 or 24 hours to make a choice. So that's basically how everything works and the benefits of it. Now, how do you actually run it? So again, The instrument is basically going to be run anywhere. There are little caveats, hence the asterisks. There are certain requirements that have come either between the power and the location within a lab and how that's handled. It can be mobile in the sense that you can't have it on the back of a truck just driving it around. It does have to be in a stable location. Again, run by almost anyone. Once you go through the process, it's very simple to run, and you'll see how easy it is and how you don't need to be a trained microbiologist or scientist for it. Again, since the machine's doing all the work for you, you don't have to worry about having people coming in at any time of the day that the results will be coming out, or for holidays or anything like that. And again, it can be set up to either have email or not, and so they get results as soon as they're available. All right. So the cartridges, again, there are 4 different types of media that can be run in here. There is this 5th cartridge at the end here, this black one, and that's actually the optical validation cartridge that comes with the instrument. That will be used to verify that the instrument is running properly optically. Again, there's the B4, which allows you to do 4 samples, and the B16 up to 16. And again, it can do different incubations within that one unit. So if you were to be testing for coliforms and E. coli, you could also be testing enterococci at the same time. Again, there's always little caveats to some of these. So there are 4 different segments of 4. So if you start running an E. coli coliform test here, this set of 4 can only run that sample. But next door, if you want to throw an enterococci, you could do that. So it allows for multiple tests to be run at the same time. And again, the instrument is incubating the entire time, so you don't have to worry about incubation, and it's also reading constantly. So therefore, as soon as it sees that signal and deems it's a positive, it will run the test up until that point. So one thing that I always like about our tests is that they're very simple to run. I always say that if you can make Kool-Aid, then you can run our tests, because basically what it is, it's powder, water mixed together, and that's about it. It's no different for Tecta. Again, the cartridge contains the reagent, so there's nothing to do there. You just need to pop the top open, pour in your 100 ml of sample, either by using a pre-measured amount or by using the line that is on the cartridge. This red line here is just drawn in there for kind of visual purposes. It does not exist. But basically pour up to here, and then snap the top of the cartridge closed, make sure it's completely sealed, and then basically just gently swirling the sample. I will say that we definitely stress that it has to be gently swirled, because if you shake it and create a lot of foam, that can cause some sample to bubble out the top, which we don't want to do. So very easily, just take that sample and swirl it, and then you can set it down for the moment as you do the next part and let it completely dissolve. And for the next part is the inputting of the information into the Tecta itself. So there are several different fields to fill in, including sample ID. The mode standard is the one that we're talking about now. The test, and so this is where you decide which reagent you're using, and this will determine what incubation that machine is going to do in that section. And then finally, the type of water. There's drinking, ambient, and wastewater that can be placed into here. And you can actually do a dilution right here. You can factor in if you did a 1-to-1 dilution because you had to do it for the sample for whatever reason. There's a drop down here, and you can choose different dilutions, and it will calculate all of that for you in the final result. Finally, here you can see that chamber 1 has been chosen. So once all the information is completed and ready to go, you hit click add, and now that chamber is now set for that sample. At this point, if you're just doing one sample, you can go through the next part. If you have multiple samples that you're running, you can go ahead and go through this whole process for multiple chambers and just have your samples ready to go and make sure that you have them going into the right spot. And then to actually put it into the machine, it's very simple. Pop the lid open, and then take your cartridge and put it into the corresponding chamber of the unit that you have set up into the previous data file. If you just do one, throw it in there, close the lid, and immediately what the first thing is going to happen is that this little blue bar here is going to start showing up. And what the unit is doing is it's looking to make sure that you've actually put something in that chamber that you've set up. And then at that point, it starts the incubation and starts reading all at the same time right away. If you have multiple samples in here, you'll see all the different ones light up as it goes through them all. Now, if you put in one and then either immediately you have more samples or several hours later you get more samples come in, you can add them to this even though it's already started with this one set. Basically, you go through that same whole process of putting in information into chambers that are empty. Then you would open up the top and put them in and then close it and then continue on. I will say that if you already have samples running and you open it up to put more samples in, there will be a timer that will start because you can only have the lid open for 3 minutes before the machine will void all samples. So thankfully, there are plenty of visual and audio cues to let you know that you've left it open and that you need to have it closed before that 3 minutes is up. So once it's in there, you'll basically see the cartridge that's being tested. It will kind of light up. Each of the cartridge chambers is labeled 1 through 16 in this case, because this is the B16. And then they'll have different colors. So if you see a red icon, then that means the sample has run through its test, it's complete, and it's positive. In this case, red is bad because you don't want to find these bacteria. So it turns red saying it's complete, I found it. Underneath the number, you can see a very small time. That's basically when the test is completed and when it found a positive. If you see yellow, that means the test is in process, it's not complete yet. The time that's found in there is where it's at within its 18 hours. Green means it's complete and no target bacteria are found. So your sample is good. Again, that time under there will be the endpoint, in this case 18 hours, because it didn't find anything. And if you had multiple samples going on, you would have, of course, all of them lit up differently depending on where they're at in the process. So get all of that. Next thing is, of course, to read the results. So this is a screenshot of what you would actually see on the unit for a completed test. It has all the information that you put into it. So your sample ID, when the sample was started, the water type you've chosen. Again, 18 hours is the full duration of the test. If it's negative, so it's an 18-hour test. If you added a dilution, that would show up here. About 100 mLs means the number of amount of mils of sample. The target temperature, so this is the ECTC test, so 35. And then during the incubation time, it's going to record several times during that incubation and collect that information and present it on to the report. Because if you do incubators in your lab, you're supposed to look at the temperature at least twice a day with 4 hours in between. So Tecta Instrument is doing that for you and collecting that information as it collects. So actually one thing I'll point out here that I didn't is that in this chamber here where we have one going, if you look, there's a number here in the middle. That's basically the average temperature for that incubation section. So that is a visual cue telling you where it's at. But again, the actual report will give you several data points. And then finally, the bottom is the actual results. So this is an E. coli result of a present. It'll give the time of when it turned positive and what that associated value is quantity-wise. So in this case, it went for almost 18 hours, result of 1. It's also coliform positive, and it's present here. Time of about 16 hours, 24 minutes, which gives you a result of about 15. So this is what you would actually see on the unit itself if you were to look at the results for your sample. And this is a positive, and this would be for a negative test. So all the information is the same up here, but you can see here, E. coli is absent, coliforms is absent, and it has elapsed times of 18 hours. So that way you have proof that it ran for the full 18 hours and it didn't find any target bacteria. So this is what you'd actually see on the unit itself if you were looking at the results. If you do have it set up so it's also sending you out an email, you'll receive an email that will have the same information. You'll see a subject with the result, all the same information here, just in the email format. And actually attached to it will be a CSV file that you can then download and put into your data system. And again, if you don't have the email set up and you collect all this information, it can be pulled from the system just using a USB plugged into the unit and then archiving that data, and then you'll have the same CSV files and other information that you can then pull off and store into your own LIMS system. So some other general notes, just like most enzymatic tests, it's a confirmed test, so there's no confirmations required. Results are final, nothing else needs to be done. As mentioned, that black validation cartridge should be used monthly in order to verify that there's nothing going wrong with the optics system of the unit. And then the cartridge is certified for 2 years, and so every 2 years it should be recertified to verify that it's still at the right fluorescence for the test. And then finally, with any test, dispose of according to your local regulations. So, I mean, again, pretty simple on how to use it, like most of the tests. Now, how we use it, again, mentioning that they are not, for the most part, used for compliance testing. It's mostly non-compliance testing. But what I can tell you is quick answers for different situations or different environments. So we talk about like construction testing or any kind of main breaks. You can quickly get a sample running. And if there is concern that something's going to turn positive, you'll get a result anywhere between 2 and 18 hours. So if there is an actual issue, you can hurry up and make a correction and move forward with that. Again, since the machine's doing all the work for you, you don't need to have plans on having people come in either on off hours or to have them be specifically in the lab to pull the results at a specific time, especially when we talk about holidays. So it makes that a little cleaner. Large cities, if you're pulling samples from across a very large geographical region and it's going to take you hours to get your sample in, again, this can be set up in a very small office space, if you can have that set up. And then you can actually just have points around a large location to have samples testing. And we do have customers in North America who do this, where their central lab will take most of the samples, but for those on the outside, they might have another location where they're just having some of the samples sent there, so that way they don't have to deal with traffic and getting things moved over to that location. Of course, you can have it near beaches, so if you're concerned about that, you don't have to worry about travel. Do it all right there at the location where it's used. That includes wastewater treatment plants. You do it right on site. And then, as we talked about, not the best image, but you know, anything that might have a color or turbidity to it, you can do it right there on the spot and test those kind of waters. Talking about staffing challenges that I think everybody's still dealing with these days, whether it becomes sick days, people retiring and not being able to fill those positions. Again, having something that is automated allows you to continue on with hopefully no breaks after you deal with all of that. And then finally, kind of like those hard to reach locations, such as mines, military locations, or cruise ships. Again, trying to get somebody to do the testing can be difficult, and getting that samples back to the locations. So one example I wanted to show, again, we have different examples of studies that have been done with different types of waters. This one's from the CEAEQ, looking at an issue where remote communities wanted to test their drinking water and didn't want to have to move the samples from a very large long distance location. So problem, you know, no local laboratory, no trained microbiologists, and they want results quickly. So the solution was to add a B16 unit in those locations and have the people themselves do the testing in those locations. So done in a couple phases. The first phase looked at both a artificially contaminated sample where they're taking clean water and adding bacteria to it, and then naturally contaminated samples and having those run on both the Tecta and on the reference method. And the results from that testing showed that Tecta and the reference method were equivalent in PA format. Second phase, doing the same thing, but now just testing a larger volume, a larger number of samples of naturally contaminated samples. In this case, they did 58. Again, Tecta versus the reference, and 8 out of the 58 were positive, and the Tecta agreed with the reference method. All right, so to conclude, the Tecta technology is a novel approach. It is fully automated from basically start to finish when it comes to the actual testing. It's the same enzymatic technology that has been used for decades that has proven to be reliable. Its quantification is based on growth kinetics, which again, all tests deal with it in different ways, but this just takes it to a different level and how we interpret results. The partition technology of the cartridge allows for a wider variety of samples to be tested without having to do any kind of manipulation of the samples, such as dilutions. And again, kind of one of the things that we tend to hear about is people and trust in people reading results. This takes away all of that by doing it automatically with the machine. Again, data management is also very important to make sure that everything is done correctly, and that's all taken care of and recorded by the machine, so it's available either from the instrument or, again, can also be emailed directly to the user. It complements the other tests that IDEXX provides. So it's never assumed that this would replace everything that's currently being done with the more traditional DST or enzyme technology, but it is a complementary thing that can be used in a location. Again, anytime, anyone, anywhere, with some caveats. But anyway, it's a great technology, something we're proud to be working with, a couple different things, and we see it does have benefits in many different applications. And yeah, I'm going to leave it open to questions within the next few minutes of the time.
Thank you, Shawn. That was great. Applause. There you go. I had a question, and then you answered it right at the end there. I was curious about maintenance frequency. So.
Yep. Yeah, and so again, the optical validation should be done on a monthly basis, but there are other things that can be done, and I'm not getting into the part of it, but there are service plans that can be worked with as well.
I also had a question, but you answered it. Luckily, I had many questions. I may have missed this at the beginning because I was shamefully late, but so there's 16 compartments, yep. And you mentioned, can they all be programmed? Like if you're just doing E. coli, can all 16 of them run E. coli?
Yes.
Okay.
Yep.
But you can tune them to another test. They have the capacity to all 4 quadrants run different tests.
Correct.
Okay. Thank you.
Yep, with one caveat that they have to be very similar in temperature.
Yeah.
So 35 and 41 degree temperature tests can be run at the same time, or 41 and the 44 and a half. You can't do the 35 and the 44 and a half at the same time. That's basically the only caveat.
Okay, one more question real quick.
Yeah.
The jars?
Cartridge.
Cartridge.
Yep.
They're disposable, yes?
Correct.
They're single use?
Yep.
Yeah, thank you.
Yep.
Are they recyclable?
They are not.
Okay, thank you.
Yeah.
Had to ask. Any more questions from the crowd? Oh, one back here. Almost missed you.
I might have missed this, but just wanted to confirm, so you can use it for kind of internal monitoring, but it's not accredited for external if we are reporting for compliance. Is that correct?
Correct.
Okay.
We're taking orders today, so.
Oh, and we have an online question. We got some time. They're using a handle, so I can't read the name, but the question is, are you in the process of looking at applying for accreditation with the MOE here in BC?
So that's a great question, and I do work with regulatory a bit, but I can't exactly say where we're working on and where things are at right now. But we are always looking at what makes sense and try to get approvals for the technologies so that people can actually use it for compliance testing. So.
I think the challenge we run into in the operations side of things is that our regulators like to see us use a third-party independent tester.
Okay.
So that's always kind of our challenges. I don't know if there's many. Well, I'm sure Metro and some of the bigger plants have accredited labs, but I know most of the smaller facilities, you know, it's not worth the investment to have an accredited lab with a lab tech that meets the bar with that. But very useful in a pinch when your lab's closed for sure, or if you're in a remote site, obviously. All right, we have a 15-minute break before the next presentation. Let's give Shane a warm round of applause again.